Alright, hello everyone, welcome back to the channel. I know it's been a while since I've posted anything educational, but that's not the point here. The point here is that I have very little time. I have to get right into this right away because I have literally 45 minutes to study all of these slides. Literally, you can see right here, it's 130 slides. And I just want to show you my process on how I learn 130 slides in exactly 45 minutes because I have somewhere to be at 7 o'clock. So let's get right into it. So we have all these slides with all of this information. And we also have my notes here that I have on my iPad. So if you look at this, I downloaded this program called like Reflector 4, and you can see the stuff I'm doing on my iPad, like through like AirPlay. So these are a bunch of notes that I took during class. But in my opinion, these are kind of garbage. So I'm going to start over here and show you exactly how to review for biology, like, or honestly any subject, like even physics, even like chemistry or like organic chemistry, like all of these things, because I'm taking those two classes right now too, and I use the same thing to study all those. So I'm going to start at the very beginning. And just maybe put it over here so you can see like the scroll bar. So I can see the scroll bar. So membranes and transport. So before I even start, I'm thinking about membranes and transport. So what do I know about membranes? Well, I know that cells have membranes, kind of like this blue thing over here. And I know that transport is like things going in between like cells. Like maybe something goes into the cell, something goes out. So you can see right away that I'm thinking about, well, what this topic is about. I'm not just like going to the first slide already. It's like, okay, you have a topic and you have to think about how this topic actually matters to you, because maybe you're taking this class for an A plus, or maybe you're taking this class because you genuinely need this information at some point in your life. So you really need to think about learning it and learning it in like a way that makes sense and matters, because that's how things stick. So learning objectives, I'm just gonna skip this. I don't have time. Like this is going over the stuff that we're gonna learn about in slides. Usually a great place to make connections, and a great place to review. But I'm learning this for the first time, so I'm just gonna go right into the content. So this is a lot of content right here. Before I even read anything, I'm thinking, all right, plasma membrane, nourishment, protection, and border. So what I know about the cells is that it protects the cell, and it's literally the border of a cell, but I actually didn't know it can nourish the cell. So I'm going to look here and look for something that says about nourishment. So okay, entry of nutrients and exit of waste. So now I learned how like a membrane actually nourishes the cell. It's like, I don't believe, as of what I've learned so far, that the cell actually creates nutrients, but it helps in order to help like do nutrients for the cell. So yeah, there's a lot of things I don't really understand, like what aggregates mean. So I'm just gonna skip that, because if I can't like know how to think about it, then I'm gonna have to just come back to this later. So yeah, let's just keep on moving on as quickly as possible. It's not about getting all the information that we can. It's about just like creating a framework as best as we can. Clinical importance of membrane protein ion channels, cystic fibrosis. So this is some sort of disease. So I'm just going to get the main idea of what's going on here. So I see this picture where chloride ions aren't able to move through, but over here in cystic fibrosis, these chloride ions are not going to go through. I know that chlorine is important for sending information. What is chlorine important for? All right, so they didn't really tell me what chlorine is important for, but I'm just going to just replace it with nutrients. If nutrients are not able to leave the cell, that's very bad, and thus it looks like it's specifically lungs, because I'm thinking about cystic fibrosis. Well, I don't see any word that relates to lungs there, but Oh, it affects the pancreas and other organs as well. So, and it's genetic. Okay, well, that's a lot of information, and I don't think I'm going to be able to remember that. But this protein, it's to do specifically with chlorine ions out of epithelial cells. Oh, from different classes, I know that epithelial cells have a lot to do with mucus, and secretion. So I know that this is specifically to do with that. So using information that I know, I'll be able to create stuff. So I would pull out my notes and like write stuff, but I'm actually going to go a little bit more and see what I can learn, so I can just like connect all these ideas together. So we just talked about a disease that has to do with the membrane, and like how the membrane is important for secretion and the overall health of an organism, especially specifically in the liver. I'm sorry, the liver and the lungs, the pancreas. Okay, the pancreas. So I'm just gonna keep going. So more structure and function. We're gonna look at this. Okay, so to be honest, like this is kind of stuff I already know, so we're just gonna keep going. Sterols like cholesterol, and lipids like phospholipids. And again, the goal here is speed. If you see, it's already 6:17. I have to do all these slides by 7 o'clock, so I'm really rushing through this and just learning as much as I can in this little amount of time. This is the kind of stuff I already know, so we're just going to keep going. All right, so this took me a way too long to read one slide. I would warn you guys not to speed read. So anything that you do, you have to do with intention. You can't just be reading randomly. You have to have something in your mind that you're trying to accomplish when you do anything. And the same goes for reading. When you're just looking at a slide and just like looking over the words, it's not going to stick with you unless you're actively thinking about it. And what's more, right? It's fun. It's going to be more fun for you if you're actually engaged with the material. So let's keep going. Okay, so I felt like I'm just done a good chunk of learning right now, because the next slide is going to be about history. So what I'm going to do is that I'm going to consolidate my learning onto my notes. And my notes are not going to be any sort of linear fashion, kind of like bullet points, this. It's going to be really a reflection of what I know about the topic as a whole. So I'm going to start off right away with what membranes are, right? So membranes divide cells. So I'm going to draw a cell, and I'm going to draw a plasma membrane like this. And I know that in a cell, outside of the cell, we have the extracellular matrix, which is a different topic entirely in biology. And in the inside, we have the cytosol. So in the fluid membrane, so I'm going to write fluid right here so I can get back to it. But I'm just going to write fluid right here because it has some importance to it. So in the fluid membrane, we also have like proteins. So proteins can transport stuff. So proteins can have, so cells can have little ions in it, or a lot of stuff. Let's just say this is ions, but ions and then etc. So they can leave through proteins, and they can also go in through proteins. And this certain protein, I know there are different types, but that's not what we learned about right now. So I'm just gonna keep on going. So this fluid part right here is important because this is sort of like wavy, like it's fluid, like the semipermeable, sorry, the fluid membrane, literally the bilipid layer, is fluid, and that's because of these unsaturated fatty acid tails. This is going to be a glycerol. I know this from background. Glycerol. So these are unsaturated fatty acid tails, and when it freezes, so let's say that I'm going to draw another line here, and I'm going to draw a snowflake. And a snowflake has, it's cold. It's cold. And the snowflake, when it just represents coldness, when the bilipid layer actually is cold, I know that it can become more solid and more like hard jelly-like structure. Hard. So this being hard actually reminds me of something else I learned, is that this layer here also has little things called it has sterols, and these sterols are actually cholesterols. So cholesterols, I'm going to make another layer right here, cholesterol. So you even notice I've been really finicky about the colors. I should probably stop being finicky about colors or just get faster at switching between colors. That's kind of the honest truth. Okay, so cholesterol, it maintains the like the fluidity of the membrane. So cholesterol makes it's just there. So cholesterol can like when it's cold, it increases fluidity. So I'm just gonna write snowflake increases fluidity like this, and then I'm gonna write if it's hot. So I'm gonna draw fire. It decreases fluidity. So overall, this tells me that cholesterol is kind of like a buffer for the membrane. So this is what I remember. I wonder if there's anything that I missed. Oh yeah, I learned about that that disease. So if these ions are going to be, if there are Cl minus, and this is an epithelial cell, you have something called that disease. It's like I forgot the name. So epithelial cells, and these things have mucus on the outside. So yeah, they got mucus out here. Mucus. And this stops chloride ions from exiting, and it is really bad for cell. Bad. Now, I think that's a lot of what I remember. So I'm just gonna go through it really quick. Oh, empathic. Okay, without even looking at this, I'm just gonna write empathic. I remember what it means because it's. Where can I put this? So where can I put empathic where it connects to the idea? I'm just gonna draw a line just like this, and I'm gonna write empathic. thick, and I'm gonna say for lipids and for proteins. And I'm just gonna draw a diagram here that shows hydrophobic, So I think I'm spending way too much time here. So my goal was to end at 7, but I've spent way too much time on just this first part. So instead, I'm just gonna pick up the speed. We have a lot of material to cover, so I'm just gonna keep going. If your professor really needed you to know these names, I would recommend that you put them into Anki cards. Because that's like straight memorization stuff. So I'm looking at this right here, and I know that this model is wrong. I just know it. So now I'm questioning myself in my head without even writing my notes. Why is this model wrong? It's because, well, I know that proteins are actually embedded in the bilayer because proteins are amphipathic, and they have hydrophilic ends on either side, and the center is hydrophobic, just like the lipid bilayer. So this model is completely wrong, but this is going to be more sticky to me because now I know why it's wrong. I'm going to pick up the space a lot, like tenfold. Because this is stuff that's very easy. So at first learning was very slow, but then I can see that it's a lot of review, so learning can be very fast. It's like it's the snowball effect. Whenever you know more about a thing, it's easier to learn. And I'm just nodding my head like, yeah, I know what's going on here. Okay, whenever a slide looks like this and there's a lot of long words, I don't think you just have to know this. So I'm just going to get the main idea, distribution of phospholipids, and I see there's different types of phospholipids. So I'm just like, cool, there's different types. Cool, let's move on. I'm just looking at the significance, but yeah, that's it. Okay, and for things like this, it looks like there's 3 rules I have to memorize. Instead of trying to memorize it word for word, I'm just going to be like, why does this matter? Length of hydrocarbon tails. That kind of makes sense because if I'm going to draw right here, if I have like a bilipid layer, if my tails are really long, compared to, really short tails, of course it's easier for this one for anything to pass through like that. So that just kind of makes sense. So on the test, I can just rederive that. Let's similarly go through what the other things are here. So saturation state of the hydrocarbon tails. Yeah, so if you have like this is going to be a lot harder to pass through than, something that's like this, where it's like, because of these kinks right here, things can go through much easier. Things can't go through right there. So that just makes sense. And then presence of cholesterol. Yeah, if there's more cholesterol like in here, there's more cholesterol, then it's going to make it a lot more fluid. And if it's more fluid, then it's easier for stuff to go through. Okay, yeah, move on. See, you just gotta be like, just why stuff. And then it just makes sense. You can rederive it on the test. See, I already know this, so I'm just like going, yeah, literally. We literally do this. So, yeah, you can just keep moving faster because you just learn stuff. And this is exactly what I drew earlier, so with the kinks, it's easier for stuff to move too. Yes, Charles. So here, I'm trying to recall what it's called when there's 2 different charges. Oh, it's just polar. Okay, never mind. Amphipathic is when it's polar on one end and polar on the other end, but in the middle it's a different one. So yeah. Maintain memory fluidity. If I had more time, I would go through each of these questions and wonder why they're wrong. And I would go to the correct answer and look at why it's correct. But I don't have the time to do that right now. For test review, however, that is something I will definitely do to understand everything. This is a secondary thing from different. So I'm thinking about different units too, not only just the unit I'm learning about right now, but units that I've learned about in the past. That's you can make those connections for the final test. So I see this amphipathic. So here, I just spent way too long rereading this sentence, wondering, well, not even wondering, just trying to understand it, when I really had another question in my head. So I had a question on why my proteins would need to hang out on the top here. So this sentence was essentially saying that these create covalent bonds with the hydrophilic heads on this side, and that's why we keep them there. But the whole time I was just thinking about like why they need to be on the outside when I should have just like skipped this. So now I'm gonna go answer that question. Why do they need to be on the outside? Does this tell me? No, this doesn't tell me why. So I might just Google it because. Oh, okay, so here are different functions. So functions determine structure. I know that. So maybe that'll tell me why some proteins are on the outside, and it's not over here. So it's either not on the test, so I can chill, or it just doesn't matter. I mean, I'm sure it does matter because biology is like a very intricate subject, but looks like they're not going to tell me why some proteins. Oh, membrane cover. Okay, so here. So essentially what I'm practicing right now is something called inquiry-based learning, where you have a question and you seek for that question to get answered. Because you, okay, glycoproteins. Has stabilizing and protective functions. Okay, this makes sense. Now, as I was saying, with inquiry-based learning, whenever you learn fueled by your curiosity, it's much that information that you'll learn by answering that question is going to stick much easier than just shoving information into your head and hoping that it stays there for test day. So now that I know, whenever I think about, transmembrane proteins, membrane proteins, transmembrane proteins is what? Trans is through. Unless transmembrane. I'm gonna look it up. Is integral. Okay, so integral proteins are transmembrane. So when I think about a membrane protein that just stays on the top, I'm always going to remember that it's going to have something to do with structure or protection, because that's why it's going to be out there. So I'm never going to forget that. So I'm back to where I was skipping. I feel like I'm rapping. I'm just talking so fast. I have no time. No, I went back because I'm wondering, like, why is it linked to one? What does it matter? Because if I don't know how it matters, like, I don't think they might even test it on the test, because, like, how can I know how that matters? Why do we need that? Why does the body need that? Transfer it. This makes sense. Yeah, makes sense. So transfer that these names make sense. Receptor, I'm guessing it takes something in, like extracellular molecules or whatever. Hormones. So hormones from other cells. Okay, makes sense. Cell adhesions to stick to other cells. Binding receptors. Okay. So these just make sense. They're 4 different functions of different things. So now I bet we're going to see. This is a transport. Yeah. So this looks like this is a metabotropic sort of protein. Glycoproteins, I'm going to go back here because I got curious to read about what significance of glycoproteins have. Oh wait, no, it's down. Okay, so we're going to learn about glycoproteins a bit more down. Yeah. So it looks like this is non-covalently. Okay, so probably thinking about van der Waals. Okay. If something is circled, you might really need to know that, so. Oh, so these oligosaccharides really remind me of what I've seen over here, right here. So this is probably an oligosaccharide that's involved in cell recognition. Addition of carbon. Okay, so I got this wrong, so I'm just gonna go back in because it looks like we need to know that. Oh, it's a bolded word. I can't believe I missed that. So let's actually slow down just for a minute and read this. So I'm still confused. So I'm just going to skip this and keep learning more about like glycolipids and glycoproteins. But all I should know for now is that it's the addition of a carbon to a protein or a lipid. So it's like probably the process in which it is actually joined by, you know, dehydration synthesis, where they remove water and two monomers are joined chemically. So it looks like glycolipids and glycoproteins are actually slightly different from the membrane proteins that I read earlier. So this is actually an error I had in my understanding, and now I'm fixing it very urgently and very quickly and over in my head. These 15 sugar units, again, oligodendrocytes, and oligo. So right here, it looks like we finished carbohydrates and like proteins in the area, and we've gone on to functions. So again, this is another good point in which I would go to my notes and recall what I've learned, but no time. In an optimal case scenario, I would do that, but let's keep going. See, you really want to be what I'm prioritizing right here because I just spent like 30 seconds rethinking my priorities. My priorities here is not actually the content like review or like looking at first time. My priority here really is to actually take practice problems and like to go to be tested on this material again. So what I'm actually going to do is that since I've already gone through most of this in class and I'm not generally really confused on these topics, I'm just gonna go through this again because the lecture really went through all this whole entire thing really quickly in class, and there's a bunch of, you can see, pumps and transports here that I really need to understand and know. And since I'm more interested in learning about these, I'm going to spend my time learning about this. So exocytosis, endocytosis, and there's a lot of different proteins, like transport proteins. And this is where I'm going to spend my mental resources on, because tonicity and osmolarity, those are pretty easy topics, but channeled proteins facilitated ion channels. I believe that I can make a grouping system for all of these transport proteins and to really understand them better. So it really starts here: osmosis, water, okay, aquaporins. So I'm just gonna write down on my thing here. So I'm gonna have all of my membrane. Actually, let me bring this closer. I'm gonna have to sort this into my mind map later because all information connects. But let's just have proteins, transport proteins. I mean, all of this here. This right here is a protein. So transport proteins. So this is what makes it semi-permeable, which makes sense. So here we have so we see over here an aquaporin allowing water molecules to go through. And this is blue is a protein. So here we have our first type of diffusion, facilitated. So facilitated, that's definitely something that reminds me of that's something that uses energy. So oh, actually it doesn't. So I was wrong. So I'm going to fix that very, very strongly and quickly. Here it says that they move according to their concentration, but their diffusion is facilitated by proteins. So it's kind of like an aquaporin. So this is going to be, I'm going to write here that this is a new type of diffusion. So facilitated diffusion. So I'm going to write specifically with gradient lipid layer don't like, and I'm going to give an example of in aquaporin. Because aquaporins are important. This is terrible spelling, but it doesn't matter. Only I need to understand it, not you guys. Aquaporins are really important. So, okay. I understand the facilitated fusion. So transport proteins in facilitated fusion. So now we have to look at some examples. So aquaporin, like I guessed, ion channels facilitate all these tiny ions to ions. Or I know, as a matter of fact, of having taking a couple of neuroscience classes, that these have a really tough time going across the membrane. In fact, nearly impossible. So ion channels. So I'm gonna write on my notes. I'm not gonna build it, but ion channels, which makes sense. Okay, Gata channels, which would be an open. So now this is a different type of facilitated diffusion. So I'm gonna make another branch from here. So here we have another branch, and we call it ion. Gated channels. I gotta catch myself. Gated. Mine's gonna be active during this process. Gated channels. Okay. So it looks like this right here, ion channels right here, is actually very important. So it's gonna get its own category. I have a question. So here I see that it needs to be opened by some sort of ligand or some sort of neurotransmitter. So does all ion channels have a sort of ligand that needs to use? This is my question that I need to be answered. Oh, so I see there's different types here. Now I see that there is a type of ion channel that is ligand gated, but there's also other types of ligand like ion channels that's always open, and this reminds me of leak channels. And this is voltage gated, which is determined by the electrical gradient across the membranes by certain charged things such as ions or membranes that can be positively negatively charged inside or outside. Anyway, mechanically gated with pressure, I see. So something, do they talk about mechanical sense? I'm more interested, curious about mechanical force on the cell, mechanical sensitive ion channels. Okay, so it's coming up really close to my time where it's 7 and I have to leave, but I'm just gonna end off right here. At least, maybe I'll pick up this recording after my meeting. So I'm just gonna write out the 4 different types of analog channels: mech, electro, weak, and voltage. And the relationship between these two is that this one's to do with charge. Okay, now I'm confused about voltage. So I'm comparing these two that are very similar. Ligand. Oh, okay. So voltage and electrical is actually the same. So I was mistaken. So this is actually the same. That's why I was very confused. So this is actually going to be voltage right here. Voltage. And this is going to be, without looking at the slides, it's going to be ligand gated. Ligand. And this has like some sort of gate, and it needs a key. It needs a key to open. So the key can be neurotransmitter or whatever. So, okay. This has been a pretty good session. I will continue this because there's a lot of slides left. And honestly, look at the recording, it's a little less than an hour. What I've actually spent time on learning, I have with like a very, very high retention rate, I can say. And as you can see, my notes, very, very different, very not focused on trying to write down all this information, but really focused on trying to understand the story of what's behind all of this biology, so that whenever I need the information, I can recall it through this web of this pool of information that I've built for myself, so that I can access this. Honestly, I'm just gonna end the video here and just send a quick, fast little image or mind map that I make at the end, and maybe just review what I've learned at the end. But I have to go get a drink of water before my meeting and take a small little breather before 7 p.m. So I'll conclude my thoughts in just a little bit. See ya. Alrighty, everyone. So it's been a while. It's been a little bit of a while since that last recording. Since then, I've learned a lot, such as whenever I was shaking the table, these markers were moving, and it was like making the entire recording really annoying. So I learned a little bit. I also learned I said I'd go over the transport rib proteins, and I did. And I fully understand it. As you can see here, there's a lot of concepts going on, like this idea called facilitated diffusion. This is an idea called a gated channel, and an idea of active transport alongside 3 different types of active transport. And then within the active transport, there's different types of, like, difference between primary and secondary. So what my goal was for all of these ideas was to just simplify and connect, simplify and make sense of it. And I could always redirect this later. And this, this uniporter, this type of transport member, this type of transport protein, there's an ion version, and then there's a hydrogen pump version. And that every single one has like examples, but I just remember them. And it's really easy to remember when I'm just able to categorize them. So this is my strategy, and it saves a lot of time. Now for me, whenever it comes to studying, it's not really my study methods that are dog, it's just like my discipline at this point. So we're working on that. So for this next part of the video, I want to walk you through my multiple choice, my practice side of learning. And this is where most of the learning happens. And it's not necessarily taking the questions that make you learn, but rather the revision part of it. And you'll see that process really in depth here in this video. So let's just start. So this is macromolecules. This has nothing to do with this was membrane stuff, so that's different. This is macromolecules. We're talking about carbs, lipids, proteins, nucleic acids. Okay, so we'll just start off right away. Which of the following hydrocarbons is false? So I'm gonna guess C, process of elimination. A and B. D, I think of benzene, so it's C. Now this was actually kind of annoying, because it was a bit tricky in terms of hydrogens. So backbone, the carbon backbone is, I know A is wrong, right? But that's not my intention here. My intention here is to try to remember what the heck a carbon backbone is. And from my knowledge, I'm guessing it's just any sort of organic molecule when the C's are all lined up together. The carbons, where everything is branched off of, is called the backbone. But watch this. I'm actually going to make a note of this. So I pull up, let's see here. I'm going to pull up my device here. Where is it? Where are you at, you naughty boy? No, actually, where is it though? It was like right there. Did it stop recording? No, it's still there. It's like right here. I can see it. Where'd it go? It's like right there. Come on, show yourself. Move to new desktop. New desktop 2. Where is it? What the heck? Okay, well, I just closed that. Sorry about this. Oh, this is mad goofy. Oh, okay, now I have to reconnect this. Oh man. Hopefully I remember to edit this out. If I don't, that's a massive L for you guys. So yeah, just give me a second. Okay, looks like we're back. Are we back? Yeah, I think we're back. Okay, so cells. So here I have my cell notes. Kind of all over the place. Actually, no. Yeah, I'm not looking for my cells. I want my macromolecule notes. This is what we want. Okay, so I'm going to use this space right here for my multiple choice practice. Alright, so here we go. I'm going to write down a couple terms that I want to review for later. So here I know I want to practice what is the carbon backbone. Carbon backbone. So let's take apart this question. By the way, I haven't even. Okay, well, I was right, but I didn't mean to look at the answer. I was literally about to say I haven't looked at the answer yet, but okay, whatever, that's fine. I'm really looking for what I don't know that I have a doubt about. And the reason I knew it was C, by the way, is because when you think of a C, here, let me just show you, like right here, right? When I think of a C, it can have 4 bonds like this. So if carbon ever has a triple bond, it can only have one more bond here, and like this could be a hydrogen, this could be maybe it's a carbon, like another carbon. But like when I think of a backbone, like having a triple bond would simply not work logistically. So that is why I was able to piece together that it was C. Okay, let's just move on. Save some time, because time is also a priority. So matching. Interesting choice. I mean, interesting question. So we actually like do some mental work. So lipids are a diverse group. What are the groups for this? How can we match these together? I'm actually I've never seen this before, so this is all very new to me. Like I'm just doing it. Okay. This looks like a protein. Lipid. Nucleic acid, carbohydrates, proteins. Oh, these are the groups, and then we match them. Oh, gotcha. So this is for lipids, this is for nucleic acid, this is for carbohydrate. Okay, gotcha. Okay, cool. Is there anything here that I can learn just by... Is this a ribose or deoxyribose? Well, pause. Ribose has... Ribose is a part of a nucleic acid, so like you can't really say off of this. This looks like glucose. So I'm not gonna make up questions there. Whatever, let's just keep going. Actually, no, let's see. Transmit? Where do nucleic acids transmit? Is it like gamete cells, like diverse hydrophobic? I mean, these look pretty hydrophobic to me. I'm just kidding. Fuel, diverse structures. Okay, cool. It's designed to use micro. So it's false. Freezing, I don't know about. Toxic chemicals. So D is fine. A is fine. Toxic chemicals. See, I remember that the water turned interesting colors, but now boiling electrical charges. This sounds right to me. Freezing. I don't remember freezing, but let's just check this image since they provided it so graciously to us. Electrode, spark discharge, gases, condenser, vacuum, water, boiling, water droplets, water containing. Okay, so ammonia, methane, water vapor. I mean, I wouldn't be surprised if toxic gases reared its ugly head. Freezing. There's no freezing. I'm gonna go with B, simply because of freezing. Okay, this is just like testing strategies, but it's not really the point of this like stream, but like... Okay, B, freezing. Yeah, there's no freezing. What can I learn from this? There are toxic chemicals. So I'm gonna open this up and just... what's going on? Whoa. Anyway. Miller toxic chemicals. Experiment. So you can see I'm just going along. Not even the questions I get right. It's more about what did I miss? What are the gaps in my understanding? Okay. Fill in the blanks. Micromolecules with carbonyl groups are major building blocks of and participate in the What kind of question is this? Okay, whatever. So this is a hydroxyl. Major classes. Oh shoot. Okay, see, this I never bothered to memorize. I just know this is a hydroxyl because like, OChem, I guess. Major building blocks of isomers supplying... Oh, damn. I actually... Dang. Okay, this is... Yeah, this is bad. Okay, let's check. Let's see. So there's an alcohol. Carbohydrates. energy for cellular activities. OC. So how? I'm curious. Like, how does this provide energy? And I'm just gonna take this as alcohol, I guess. So ethyl alcohol. And this is energy here. And I know these can be isomers, like structural isomers. Okay, now I just mentioned something called a structural isomer. So watch this. I'm actually gonna go and look at isomers again. Just because I'm curious, to have that curiosity ready, and I know that isomers was going to be relevant to the exam, I'm just going to go take a look at isomers right now. I'm thinking about chirality from OChem. I'm struggling to think about the relevance in names between aldehyde and ketone. Like, I can see the ketones on the second carbon and the aldehydes on the first, but is that it? Like, how do I remember these names? The fact that I'm stuck with, like, thinking about how to memorize this is kind of a red flag. That means there's something going on here that I'm not able to understand. So I'm just gonna go with this. This is an A, and aldehydes are at the beginning of the alphabet, and so are these carboxyl chains. So they also go at the beginning of the alphabet. And ketones, well, they look like keys, like just like straight skeleton keys. So they go out. And I'm gonna move on. Here they provide energy, building blocks of carbohydrates. Didn't know that. Facilitating dissolving molecules. But now I know. And also, dehydration synthesis, yeah, obviously. Alright. There are many monosaccharides present in biological systems. Which of the following statements about these sugars is false? Glyceride has 3 carbons. Try this. Glyceraldehyde. Ribose is a compound carrying energy. Mannose has 5 carbons and functions as a fuel substance. Mannose makes up components of glycolipids and glycoproteins. Okay, so right here, very confusing. But these sugars in is false. Well, glycerol, back to like lipids, those got 3 available chains of fatty acids, so I'm going to skip that. It is a component of RNA, very questionable carrying energy. However, we got to compare these to the others. Mannose has 5 carbons. It functions as a fuel substance. Pentose has 5. Mannose, fuel substance, sugar. Alright, I'll give it that. Makes up components of glycolipids and glycoproteins. In this case, I have no idea because I haven't studied my sugars. Is it an actual thing, like, where they told us to study our sugars? Did they actually tell us to study our sugars? Because, I haven't studied my sugars. like, did they mention what oh, okay, here it is. So this does have 3. Ribose actually does have 5. And mannose actually... Okay, so they did go over this, but I just forgot it. So now, great opportunity to go back through this. Because, again, like, damn, I can't believe I missed this. So... what is the slide saying? Monosaccharides. Different categories. I know about this. Fuel substance, glycolipids, and glycoproteins. Interesting. So mannose, ribose, and glyceraldehyde. Alright, so while with this information, let's go try to attack this again. So, this is true. I don't believe it plays a part in carrying energy. Mannose has 6 carbons, so that's dangerous. Mannose, okay, this is true. I will say RNA might have something to do with energy. Oh, it's right there. Wait, how does RNA carry energy? Now I'm curious. I'm not sure what they mean by carry energy, because that's really vague. But I do know that mannose has 6 carbons, so yeah. So we learned a lot from this question. We know what ribose glyceraldehyde is, and how many does ribose have, how many this has, how many mannose has. You know, ribose is not it does carry energy, which is, I have no idea what that means, but okay. We learned that, wait, what does glyceraldehyde do? Energy yielding reactions in photosynthesis. Okay. This is DNA, this is fuel, and glycolipids and glycoproteins, which I'm sure they talk about later. No, it's the only time they talk about it. Okay, so, Oh, and this by the way, if you're curious, this is an etymology dictionary. This is like an actual... This is how dictionaries should be used. Like if you look at like Nickelodeon, right? It shows you like how the word is broken up and the definitions of this word. And it's actually so nice for understanding where words come from. Like woman. Let's see what woman. Lady. Man. Oh, it looks like this is really political. They never talked about the womb. No one said womb. Womb. Damn. Okay, anyway, we're getting distracted. But yeah. Hopefully you get to see that in action sometime soon. False saccharides. See this? There's a bunch of disaccharides in you memorize, so I'm gonna write down disaccharides. Galactose and glucose. I believe this. It's stable. Yes, I believe it's stable. Just purely from a polar sense, like just yeah, the from an organic chemistry sense as well. It's just able the electrons, if any, are able to bounce around. Once it like dehydrates and synthesis, and you get like an acid, it becomes acidic. It is bioplastic acidic bond. I just think that glucose might be a beta 1,4 linkage. So I'm gonna go with D. Okay, yeah. I think this one I just memorized it better. Beta 1,6. I believe that's for like. It's for sheets. It's for structural, something structural like chitin. So they never explained this, right? So now this opportunity. So I guessed, right? I guessed on this. So it's time to actually verify, like, and see. Okay, now what is this actually saying? So beta 1,6. First of all, let me control F4 so you can see my 1,4 linkage. Where's my 1,4 linkage? Here it is. Glycosidic linkage, and this is glucose, glucose, maltose, glucose, fructose, sucrose, and yeah, it's true. So it is called glycosidic linkage. So where's my 1,6 linkage? 1,6. I need my 1,6 linkage. Alright, so amylopectin. Yeah, this is structural type deals. And actually, beta 1,6, it only shows up here in starch. Which is very raveled together, which is interesting. So you have beta like monomers, but they become lactose, galactose, glucose, sucrose, glucose, fructose, glucose becomes sucrose. Alright, cool. So they perform a 1,4 glycosidic linkage. Animals store a polysaccharide called blank upon glucose. It's a glycogen. It's like amylopectin, but more extensively branched. Glycogen. Invertebrates store glycogen mainly in end blank walls. Breakdown of glycogen in these cells release glucose when the demand for energy increases. This is a glucagon, no. Glucagon really breaks down that stored energy for the organisms, for the organism itself. I don't know this blank, so let's just go. Liver and muscle cells. And this energy release. Okay. I need to study what the liver does. Liver function. muscle is a high G body part. Because I feel like there's a lot of everything in the muscles. There's a lot of ATP, a lot of gap junctions, a lot of dynesin, microfilaments, microtubules, kinesin, diasin, a lot of stuff in the muscles. Polysaccharides, like starch, blank is a polymer of glucose with 1416 glycosidic linkages, but the linkages and these two polymers differ. Like starch, chitin is a polymer. Chitin structural polymer of glucose. What's like starch? With beta 1 4 glycosidic linkages. These glucose monomers and cellulars are all in the B configuration, making every glucose monomer opposite. Because it's beta. It's like opposite. It's like flipped for every one. Cellulose. Where have I seen cellulose before? Is it on plants? Cellulose is on plants. Yes, it's in paper. We can't digest that. And it has the beta 1 for linkage. Alright. Yes, it does. The only thing I've seen is 1,6 amylopectin. Which was really weird. I have no idea. This is complex starch. Branched polymer. Alright, let's go to the last question for tonight. So long. Monosaccharides or disaccharides of carbon. Okay, looks like they fill this in for me. I'm gonna go back through this later. Let's just take a quick look at let's just take a look at the next question. Oh, they're really going in depth. Oh man, I should probably look at these later. Oh man, they're going really in depth into everything. Oh, wow. Okay, how much did I just like skip essentially? Okay, half of it. Yeah, I need to go through that. Two arrangements that can form in a protein's structure describe their conformation. Hydrogen bonds. What type of bond holds together the structure? Hydrogen bonds. Okay, what's the two arrangements that can form in proteins? Oh, yeah, alpha and beta. Beta pleated sheets, alpha helix. Hydrogen bonds. And what can I learn? Two arrangements that can form. Spirals, I'm just recalling, has two different formats, left hand, right hand. Beta pleated sheets also has two different types of sheets, parallel, anti-parallel. What type of bond holds together the structure? Hydrogen bonds, just in between. No van der Waals, just the hydrogen bonds, and there's no even shear too. Okay, well, it is getting really late, and my focus is dropping. So I'm just gonna take a stop here and continue later. As you can see, my study strategy, very technical, very step by step, very learning based, learning positive. this weekend has not been the most optimal weekend for my productivity. I'm working on it, so hopefully by the time I'm in med school, I'll be spending less time studying and more time on the things and family I love, people I love, and less time at my desk. So thanks for watching. Hope you gained something value out of this, and I'll be doing this a lot more often. I'm not gonna be posting these videos unlisted. Listed. This is all gonna be unlisted because I have a big YouTube channel now. I gotta keep my algorithm in mind from now on. So that's kind of the way things go. But thanks for watching, and I'll be doing this more. See ya. Bye.